Good morning, everyone. So here I am again to discuss about the PacBio sequencing. PacBio sequencing, as you know that, is a third generation sequencing. This slide is giving you an overview of the three types of sequencing, that is first generation, second generation, and third generation sequencing. And we have already discussed about the first and the second generation sequencing. The third generation sequencing involves nanopore and PacBio sequencing, and it is a smart sequencing like that of nanopore sequencing. So it does not involve sequence by synthesis. Very important, and it does not involve a very important step, that is the PCR amplification step, which introduces the bias. So what does it do? It apart from, or what is different from the second generation sequencing, is that all the third generation sequencing is that they sequence a single molecule of a DNA in a real time manner, or a single molecule of a nucleotide in a real time manner, which and a number of molecules are sequenced in a parallel fashion. As a result, a high throughput is generated. The advantage is that because of the sequencing of the single molecules, tens and kbs of fragments, they are generated on an average, which actually means that we get longer reads, which was a limitation in case of the first and the second generation sequencing, specifically the second generation sequencing. Okay, so what are the benefits of having the long reads? There are a number of benefits of having the long reads. One is that, for example, if you want to sequence genome of any organism, then since the second generation sequencing involved a large number of short reads, there were possibilities that there were certain pockets in the genome which were repetitive sequences, and there were certain pockets which were very hard to sequence. They were not able, they were not effectively sequenced by the second generation sequencing. And the first generation sequencing approaches, they were very expensive. Why? Because they involved cloning and then chromosomal walking. So that is why, keeping these drawbacks in mind, there are lots of benefits associated with the third generation sequencing, and they are an advantage over that of the second generation and the first generation sequencing. Another advantage that I have already talked about is that you can take DNA or RNA directly from a cell and sequence it directly on the sequencing platform without being the need to PCR amplify the genomic DNA. So that is a very good advantage. It does not let the PCR amplification bias creep into your sequencing technology. So talking about the PEC biotechnology, the PEC biotechnology is called as the PEC Bio Smart, that is single molecule real-time sequencing technology. What is the advantage of this technology? This technology, as I have already said, is a third generation sequencing technology involving sequencing of single molecules of the nucleotides. Okay, now another characteristic feature of smart PEC biotechnology is that it is different from nanopore. And how is it different from nanopore technology? Is that in case of PEC biosequencing, instead of nanopores, zero mode waveguides. What are zero mode waveguides? Zero mode waveguides, or ZMWs, are microfabricated nanostructures or microfabricated nanostructures which are actually holes, not wells, as in case of 454 and ion torrent. These are holes which are made on a particular surface, like that of nanopore. Their chemistry, their basis, their principle, and their composition is very different from that of the nanopore nanoholes. Okay, these are in the nanoscales only, tiny holes present on a thin surface The surface consists of an opaque material, and the opaque material is generally made up of either any metallic layers, aluminium, chromium, or gold. And this metallic surface rests on a glass substrate or a glass slide. And there is an interface of the glass and the ZMV, and that is called as the glass water interface. So there is a liquid or a solution between the glass and the metallic surface, so that the current, okay, so that liquid glass interface is there, that you have to keep in mind. Okay, now another characteristic feature of the ZMVs is that ZMVs are the structures which are having the diameter. The diameter is the crucial part of the ZMVs that you need to remember, and I will be talking about it in the next slide. So if you see, this is the flow cell of the PacBio ZMV, or the smart cell. We call it as a smart cell. On this smart cell is present, what is present on this smart cell? On this smart cell is present number of holes, and these holes, they are called the ZMVs, or zero mode waveguides. Each of the hole is open at the top and the bottom, and it rests on a metallic surface followed by the glass surface. Okay, that is the composition of the ZMVs. Now ZMVs का characteristic feature ये है that they exploit the property of the light passing through the aperture. जैसे microscope में, similar to that, the light passes through an aperture and we can control the amount of light which is passing through the aperture. Similar principle is utilized in case of PacBio sequencing. Here the difference is that the aperture of the ZMV here is fixed. Okay, that has already been defined while making the ZMVs. However, this aperture is generally smaller than the wavelength of light which is coming from the below. Generally, a laser beam comes from the below, and this wavelength of light which is coming from the down is greater than that of the pore size of the ZMV. Okay, as a result, the light is not able to pass through the waveguide or the ZMV. It is not able to pass through it. What happens then? This attenuated, it causes attenuation of the light. Attenuation मतलब वो वहां पे आके रुक जाती है, आगे नहीं जा पाती. Attenuation of the light ultimately results in the decay of light on the surface of the ZMVs. यहाँ पर ये decay होना शुरू हो जाती है. ठीक है. Now what happens then? Decay of and then this decay is exponential. You can see the scale. The decay here is in exponential manner, and this causes illumination of the lower surface of the ZMVs. ठीक है. This, so it kind of generates light on the lower part, just like a microscope. So it creates the most powerful microscope with a detection volume in zeptoliters. So a kind of area is created wherein in a small fragment, under say, I have already told you there is an opaque surface, opaque metallic surface. So however, in a restricted zone, a little amount of light is produced, which actually behaves like a microscope. It illuminates a particular region uh, like that of the microscope. Okay. However, the volume or the diameter or the surface which has been illuminated by this wavelength or this light is very, very small. Okay, the detection volume is very small. It is in zeptoliter scale, not in a very high scale. So this is the significance of this laser beam, or of the light not being able to pass through the uh, zero magnitude waveguides. Okay. Now, the another important feature is that the volume of illumination or the detection volume is so small that even the background or the surrounding neighboring molecules, they are not illuminated by the lights. So that is the beauty. So that is the beauty of this PacBio zero magnitude waveguides or ZMWs. So if we try to understand what exactly it is happening, is that there is total internal reflection is happening in this area of fluorescence. Okay, so what happens is that when the incident light reaches the glass water surface, मैंने बताया था there is a glass and then there is obviously inside the ZMVs there will be solution, so glass water's interface. So what happens is that there is total internal reflection. Okay, now what happens is there is that on the aqueous side, which side? Not on the glass side. On the aqueous side, there is exponential decay of the light. Okay, इसको स्टैंडिंग वेव बोलते हैं, जिसको एवेसेंट एवेनेसेंट वेव बोलते हैं. एवेनेसेंट वेव वो होती है विच इज नॉट एबल टू पास थ्रू एंड इट इज इन द नियर ऑलमोस्ट थम गई है, एटीन्यूएट हो गई, रुक गई है. ठीक है, सो दिस नियर फील्ड स्टैंडिंग वेव इट अंडरगोज ऑन द एक्वस साइड. रिमेंबर, अंदर की तरफ होता है, ग्लास की तरफ नहीं होता है. व्हाई? बिकॉज द मेटालिक सरफेस इज देयर ऑन द इनर साइड. Okay, so it causes exponential decay within the boundary or within a very small detection volume. ठीक है, अब इस वजह से क्या होता है, whenever there is a decay of the light in a very small volume, then there is generation of a localized oscillating electromagnetic field. Obviously light है, decay हो रही है, electromagnetic field है. What is the result? The result is that अगर इस एरिया में जो छोटा सा वॉल्यूम है जहां पर ये प्रोसेस हो रहा है, that detection, in that detection volume, if there is a fluorescent molecule, then they will be detected. Okay, and they will be excited and they will be detected. Those fluorescent molecules who are in that detection range, which is in zeptoliter scale or very less, that means less than 100 nanometers, for example, only those fluorescent molecules, they will be excited and their fluorescence will be recorded, and all other fluorescent molecule, even if they are present but they are in the background and not in that zeptoliter volume, they will not be detected even when they are present. So this is the principle of the PacBio sequencing.